Hi, my name is Abigail Nafis, and this is my research on the effects of GART knockdown on healthy versus ADSL1 mutant animals, specifically Caenorhabditis elegans, the nematode. So, the overall project goal of my lab and my personal project is to explore purine metabolism disorders and what happens when we knock down some of the genes in that pathway. So, purine metabolism is the conversion of PRPP, as you can see in the bottom left corner, PRPP to IMP, which is an energy metabolite, which will eventually be used in a lot of energy processes in the body. So what we're looking at is what happens when you knock down some of the gene expression in that pathway. What are the phenotypes we see in animals? And then how can we translate that into human research with some rare diseases that occur during purine metabolism disorders? So what I chose to do was to explore ADSL and GART. GART is involved in three of the steps, as you can see over here, so it's a pretty hefty player in purine metabolism. And ADSL is involved in two, mainly the conversion of SACAR to ACAR, but it's also involved somewhere in the salvage cycle as well. But what we hypothesized is by knocking down GART, because of its earlier function in the pathway, we could rescue some of the ADSL mutant phenotypes that we see in previous testing. So what we've seen previously is I've done a lot of semesters research on GART knockdown. And when we knock down GART expression, we see increased reversal phenotypes. So the worms spend more time in reverse, they go farther in reverse, and they reverse their movement patterns more often. What we've seen in ADSL mutants so far is they have a decreased speed, they're smaller, and they just have some fertility issues and some mobility issues. And this translates into our human counterparts also. ADSL deficiency, we have autism-like symptoms, muscle wasting, epilepsy, and GART deficiency, we see some developmental deficits as well as we see defects in people with Down syndrome. So, taking what we know, we're trying to see what happens when we knock down a prior metabolite to kind of stop that SACAR accumulation later on down the pathway. So how we did this is we took N2 animals, which are our healthy control nematodes, and we also took ADSL1 mutant animals. We grew them on GART RNAi, so they could have GART knockdown, or on EV RNAi, so they were control. So they only either had their ADSL if they were mutant, or N2, they had nothing knocked down on the EV RNAi. And then I let those animals lay eggs. The next generation, the F2 generation, we took their L4 stage, which are like teenagers, and we recorded them on a worm lab imaging software and scored the results. So one of the main things we wanted to look at was speed because of the hallmark of slower speed in ADSL mutants anyways. So I'm going to talk about why this result may not be what we think. But what I found from my batch of worms over the semester is my ADSL mutants had higher speed than the control. And when we exposed them to GAR RNAi, that phenotype was rescued. So they were able to return back to closer to the control speed that we could see. And then when we talked about the length of the animal, anytime there was an ADSL mutant, so GAR RNAi or EV RNAi, as you can see in this bottom graph, the size was not rescued. So if ADSL was knocked down, the animal was going to be smaller. So what we concluded from this is when we talked about speed, from my data that I collected over this semester, we found that GART knockdown can rescue the speed phenotype in this case. So meaning that SACAR accumulation is probably a major component in that speed phenotype we see in ADSL1 mutants. Then on the flip side, we concluded that GART knockdown can't rescue the size phenotype that we see with ADSL1 mutants, meaning that that SACAR accumulation is probably contributing to the smaller stature. It is important to talk about my possible error. Like I mentioned before, we know that ADSL1 mutants are typically slower and mine were faster. This could have happened for a couple of reasons. It could have been tracking errors on the software. Just increasing the sample size might have fixed this problem, or it could be the food that I had been maintaining my strains on. I grow them on OP50, and I know some other people in the lab have chosen other bacteria. So that might have been part of the discrepancy. But overall, the conclusion I found this semester is very exciting. We're doing a lot of studies with lomatrexol, LMX, which is a GART inhibitor, with ADSL deficiency in people. So we're trying to see if inhibiting GART could rescue those ADSL phenotypes we see in people. And this finding, especially in the speed, seeing that it does return more to the normal control, is super promising on using GART inhibitors as an eventual therapeutic in some of these purine metabolism disorders, specifically ADSL deficiency. But again, more testing definitely has to be done, maybe just increasing the sample size to confirm this data and its possible great findings. But I just like to thank Wendy Hannah Rose and everybody in my lab, and all images on this poster also were made with BioRender.